Welcome to Environmental Law Explored, a podcast series, the podcast of the American Bar Association section of Environment, Energy, and Resources. SEER is a member organization whose mission is to foster the success of a diverse community of environmental, energy, and resources lawyers, advisors, law students, and decision makers, and provide a premier forum for the exchange of ideas and information.
Hello, and thank you for joining us for our fifth episode of our 8-part podcast series focused on microplastics. It's my pleasure to talk today to two great panelists, Dr. Scott Coffin and Katie Boston, who are here to talk to us about the human health and human toxicology aspects of the microplastics issue. And so I'm going to turn it over to Scott and Katie to introduce themselves and to get us started.
Thanks, Kevin. It's a pleasure to be here. My name is Dr. Scott Coffin. I am a research scientist with the California State Water Resources Control Board, which is part of the California Environmental Protection Agency. I have a PhD in environmental toxicology and have been focused on studying the impacts of microplastics to humans and aquatic ecosystems since 2014.
Hello, everyone. My name is Katie Boston. I'm a principal scientist and risk assessor at Roux. It's an environmental consulting company that cleans up contaminated sites across the United States. I work as a human health risk assessor at Roux, quantifying risks to help clean up contaminated sites. And the other half of my time, I help quantify exposure and potential for causation in the context of litigation.
Thank you both for your time, and I look forward to the conversation over the next few minutes here. One of the things that we've learned through the first few episodes is that microplastics can mean a variety of different things to different people. And so we like to ask people at the outset, when I say microplastics, you think of what?
So I'm going to kick that to Scott because I know Scott has been working hard on a definition for microplastics as part of his work.
So scientists have been concerned about small plastic particles due to their ability to be ingested by organisms. And we came up with an arbitrary upper size limit of 5 millimeters decades ago that we have been using. This definition has been legally codified recently by the State Water Resources Control Board, where we set the upper limit at 5 millimeters and it goes all the way down to 1 nanometer, which is effectively as small as a particle could possibly be. And this definition includes all plastic polymers, as well as any polymer that has been synthetically changed from its natural state. So it's a very broad definition. There are basically any type of particle that you find in the environment that is not man-made and is organic is considered microplastics as long as it's smaller than 5 millimeters.
And just to chime in quickly and kind of bracket some of that in layman's terms, at the smaller size range, think more around the size of, a virus or something that's able to, get in and permeate your cells and, biologically interact with the human body. And then at the larger size, it's, kind of like a pea, more pea size, just for context.
That's great. I was going to ask for an example. I think I've heard also folks talk about the head of an eraser on a pencil is about the same as 5 millimeters. We're here really to talk about human health and the analysis of sort of the impacts of human health from microplastics. And so it would be great if you could kick us off by sort of laying the foundation for us. We're mostly lawyers on the audience here. Tell us what we need to know about human health risk assessment in order to be able to understand the conversation we're about to have.
Sure. So I'll kick us off on that one. And so I'm a human health risk assessor. And so when we're trying to understand potential impacts to human health, there are a couple different things we need to look at. So, as a human health risk assessor, in various contexts, we need to think about exposure first, thinking about multiple potential routes of exposure. So things like inhalation, ingestion, potentially dermal exposure. And then, that's kind of the first piece of the puzzle. And then the second piece of the puzzle is toxicity. At some dose, is there potential for, a human health impact. And so, often there's a variety, think about PFAS, there's like a whole litany of potential human health impacts that are associated with exposure. And so scientists, study human health risk by looking at scientific studies in various different contexts. So they can look at exposure to cells, they can look at exposure to laboratory animals, think rodents or primates, or you can conduct human-based epidemiological studies looking at highly exposed populations versus a control population that's unexposed, to potentially try to tease apart these issues of exposure and toxicity, what adverse health effect we're interested in. So that's kind of the way that I approach human health risk.
What do you do with it once you've got that? So how does it translate into a regulatory obligation or standard that someone's going to have to comply with?
At some point, you have to draw a arbitrary line in the sand of what you consider to be acceptable and unacceptable risk. And different jurisdictions around the world draw that line in different places. But the general consensus is that risks that are more than 1 in 1 million people are considered unacceptable. And that's the regulatory standard that we use at the US EPA and in nearly every US state for deriving risk thresholds for cancer toxicants. So for agents that specifically cause cancer. For agents that do not cause cancer or cause non-cancer health effects, we consider the unacceptable risk to be when it crosses a certain threshold of basically where you can reliably detect an effect to a whatever endpoint you're looking at. And for instance, that could be the level at which reproduction is estimated to be difficult to occur in 5% of the population. And for non-cancer toxicants, we're not looking at probabilities of how many people are going to be having that effect, whereas we are really thinking about probabilistic and statistics when we're thinking about cancer toxicants. So there's two paths that you basically have to take when you're evaluating a potential toxic agent.
Can you explain maybe that last point a little bit more in terms of, if you're not looking at probabilistic, are you looking at, you can define it exactly how many occurrences occur? What is it that you base it on if it's not probabilistic?
It's a little bit fuzzy in that effectively you take very conservative approaches at deriving a single threshold for a non-cancer toxicant. Looking at rodent studies, you will often take the first detectable dose, considered the lowest observable adverse effect level, or low AL, or you may even take a level below that where you can't see any doses. It's the first dose, it's the first dose exposed where you don't see any effects, and everything above that you see effects. And that's where you'll extrapolate from mammals to the humans, and then you can take even more conservative assumptions for exposure. Say, we almost always are trying to protect the most sensitive subpopulations. And so we'll say, take the 95th percentile for drinking water. And that is where we'll draw the line in the sand based on that toxicity threshold. And so typically these thresholds are highly protective. And it's extremely unlikely that the average person that's being exposed at that threshold is going to have any effects. And there's always a chance that if you're right on the threshold of exposure, that there's a very, very small percentage of the population that could be experiencing some adverse effects, but they're typically very mild at near the threshold dose, whereas 10 to 100 times higher of exposure, which could occur in very, very few people, may cause more significant adverse effects.
I will add too quickly that usually the regulatory values that are derived assume a lifetime of exposure. So that's something else to keep in mind. It's assuming 70 years of exposure for a resident living somewhere, or maybe 30 years of exposure if it's more of an occupational exposure setting.
In some of our future episodes, we're going to focus in on the regulatory process and really the nuts and bolts of that process that your science work is plugging into, but from that overview there, it's clear that there is a significant body of regulatory work and definitions and standard setting and the like that is a key piece of this as well. But before we turn to those episodes, you've really focused in on the two halves of the conversation, which are, exposure and toxicity. Can we start maybe with exposure, and can you give us a sense of where you see humans being exposed to microplastics?
So the data that we currently have is mostly focused on drinking water, seafood, fish tissue, salt, beer, and honey. We have a lot of information for those particular exposure routes and very little information for what's in our fruits and vegetables, our meat, other packaged goods, grains. So we have a pretty good estimate for about 20% of the average person's diet. We also have a pretty good estimate for what we're inhaling. There's been a number of studies looking at indoor and outdoor exposures to microplastics. And typically, we're finding higher exposures through inhalation than through the ingestion route of exposures. However, once the particles enter your body, it really matters how they get there. A particle that is breathed into the lungs is going to distribute through your body in completely different ways than if you ingest it. And so it's really important to separate the ingestion and the inhalation pathways when you're thinking about risks.
And then, what are we seeing in terms of the toxicity? What are the concerns that people should have about when you do ingest or inhale microplastics? What does it do to you?
And this is where we have very limited information. I recently completed a meta-analysis, an in-depth screening and prioritization of the toxicity literature with a group of experts. We found about 75 studies that have been published on the toxicity of microplastics to mammals, and about 35 of those are only looking in cells. So we have a lot of information about what could be happening at the cellular level, but it's difficult to extrapolate those impacts to a complex multi-celled organism, the whole body. For the in vivo studies, or the studies that actually expose live animals, typically rodents, we are finding consistent impacts to male reproductive endpoints. Rodents that are exposed to microplastics in drinking water or that are being fed through their food, they're finding that these particles, they spread through many organs in the body, but they also tend to accumulate in the reproductive organs, both in male and female rodents. Once they're there, they cause oxidative stress and inflammation, and this causes a cascade of processes within the organism that can impair the reproductive function. We see this in lower sperm counts. The sperm that they do produce are less mobile and less viable. They have abnormalities and deformations. And they also have lower testosterone in the male rodents. The vast majority of the reliable evidence that we have has been focused on male rodents, and particularly on this reproductive pathway. We only have 2 studies that have looked at female reproductive impacts, and they both found very consistent impacts to a single biomarker that is representative of reproductive success, anti-Mullerian hormone concentration. Like I said, it's very limited, very preliminary evidence that we have. And there's a lot of these studies they're simply not being conducted to the level of rigor that a risk assessor would want in order to set a regulatory threshold.
Is there a reason why that's the case? Like why there isn't more studying going on and at that really, high good laboratory practices level?
I think I can speak to that quickly. So, unfortunately, conducting good science takes a really long time, and microplastics also are a complex exposure media. So, you can, if you picture microplastics that you might scoop up on a beach and then, spread them out. There's a variety of different shapes and textures and colors and additives, different sizes. And so when you're trying to study that in a laboratory context, the world is your oyster. You're not just studying, what various doses of lead, and then, what is the response to lead, one exposure to, one potential toxicant. There really, microplastics is millions of different types of toxicants. And so trying to untangle that mess and quantify, actual human exposure is difficult, let alone the diversity of studies that you need to conduct to, adequately quantify the spectrum of possible exposure. So it's just going to take a long time to get the type of data that we're used to touching and feeling as risk assessors. Usually you don't want to see one study, you want multiple studies, confirming the same results and, replicating that result. So yeah, it's difficult for microplastics. I think it's going to take a while, for us to feel very comfortable.
Is there any attempt to sort of coordinate or to prioritize studying that's going on? I just think about how long it may take with, the millions of different permutations, and you could see there'd be a lot of studying going on, but everybody's doing their own little version of it, and it maybe doesn't add up to anything that you can reliably rely on. Is there any sort of effort to try and prioritize, or is there any indication that we should be categorizing microplastics in different ways?
Absolutely. So the European Union has allocated $30 million to investigating the human health effects of microplastics over the next 5 years. It's through a consortium project known as the EU Priority. It's an acronym. These projects are highly coordinated. They have a number of working groups that meet regularly to make sure that their objectives are complementary to one another and that they're not duplicating efforts. And we've learned an incredible amount about the diversity and handling this complex, tangled mess of microplastics in the past 2 years alone. We're using sophisticated statistical approaches, probabilistic methods to deal with the universe, and breaking away from thinking about categories. When you go outside and you decide what kind of clothing you want to put on before you walk outside, you look at a thermostat and it tells you the exact temperature. It'll say 75 degrees Fahrenheit, and you understand 75 means, oh, I don't need to wear a jacket. It doesn't tell you it's hot outside. And so for microplastics, we're using the same approach. We're using exact numbers. We're using exact probability distributions, and we're breaking away from categorizing in terms of binning. These size particles, for instance, these size particles in this range are this much more toxic than these size particles. This polymer is this much more toxic than this polymer. We have no indication that any one polymer is any more toxic than one another. The only indication that we have is that the size, the shape, and the density are important factors.
What do you see as the implications for arguments around causation and folks who are litigating these issues in tort cases? To the uncertainty, how does that impact that aspect of this issue?
Yeah, so I'll speak to that right now. I think, the data is really still in a nascent state, right? Where it's rapidly expanding. But unfortunately, when you're evaluating, general causation, for instance, you do look for multiple lines of evidence. So you're going to look for, some of the mechanistic data that Scott was describing earlier. You're going to look for evidence in exposed rodents also that Scott was describing earlier, maybe in primates, other sorts of laboratory animals, perhaps. But really, you also need good epidemiological evidence as well, kind of translating these exposures in a laboratory setting to actual people at exposure levels that are pertinent. So that is still a large question mark. We do have some occupational studies for synthetic textile manufacturers that show inflammatory effects. to the lungs, things like asthma, other diseases, lung inflammation. So we do have that type of evidence in an occupational setting, but I think that this is a challenging issue, when we're thinking about trying to conduct an epidemiological study. Who is the highly exposed population? Who's the unexposed population? Trying to figure, and there are ways that we can do those types of studies. There's ways epidemiologists can, control for exposed and unexposed populations, but still, it's an added layer of complexity, and that really is kind of a big missing piece right now, is good epidemiological evidence.
Where do we go from here in terms of the science development? And, this is another area where, like many environmental areas, California is really leading the way in some respects. So maybe, Scott, if you could talk a little bit about where California is, and then any other thoughts about where the science and human health impacts goes from today to the future.
So in California, we are really focusing on the exposure component. We just completed a method standardization, which allows us to go out and do some monitoring and interpret the data in a reliable way between laboratories. This is the first time that we've been able to do this based on this method standardization. So starting next year, California will issue monitoring orders to a small number of water systems that serve water to the vast majority of the state. And we'll get a good idea for the microplastics that are in the source waters that are used for drinking water, and get a handle on at least the drinking water component. We're also monitoring exposure through other routes, such as I mentioned before. We need a lot more data on what's in the majority of our diet and food, but that data is forthcoming from the research community as a whole. Apart from the exposure, there's two main aspects there's two additional lines of research that need to happen before we can derive a regulatory threshold. One is determining the effect mechanisms within the broader suite of the plastic particle universe, and narrowing down what contribution different factors have. I mentioned before size, shape, and density. We believe that those are important. There's other factors as well, such as surface charge, protein modifications on the particle, complex factors that can be teased apart with a small number of very well done studies. Once we have that, we also need a quantitative threshold. So we need a couple of good studies looking at the most sensitive human health endpoint that are conducted very well in terms of exposing the animals to known quantities of a diverse range of particles. And with those key pieces of information, we'll be ready to derive a regulatory threshold for drinking water, as well as other matrices as well.
That's exciting. And it'll be interesting to watch, both from a science perspective, but also from a regulatory perspective, and from just a person who drinks water perspective. It's great to get the sense of sort of how that is going to be developed and where we are going from here. As we wrap up today, I like to give people an opportunity, who are the panelists, to say, give us one takeaway that you really think is most important. If you wanted people to leave this conversation or this episode with one thing that you've said that really stands out, let me ask each of you, if you could, what would that one thing be?
So a lot of people may be wondering, how can I reduce my exposure to microplastics? At this point, we know that a lot of our exposure is through inhalation, and indoor air typically has 10 to 100 times higher levels than outdoor air. Most of this is through our furniture, our carpets, our rugs, even our synthetic clothing that we wear. And a very simple thing that we can all do to reduce our exposure is to install an air filter. Get an air filter for your house. It will reduce your exposure to a lot of other nasty things that you don't want to be breathing anyway that are not microplastics. The other way that you can reduce your exposure through the ingestion route of exposure is to consume less plastic packaging food. When you open a water bottle, it releases thousands of microplastics. When you tear open a plastic package with food in it, it also releases thousands of microplastics that make its way into the food, into the air. And so this is a very simple way that you can not only reduce your personal exposure, but also reduce your plastic footprint into the environment.
My quick parting thought is that, often I feel like I come into the room and then scare everyone. It's my job to scare everyone. And so I just want to, reassure people that, there are some crazy statistics flying out there about, how much microplastics we're all ingesting and inhaling. There's a large fraction of those microplastics that actually aren't able to make it into your biological systems. Yes, a fraction of them can, but just kind of rest assured that not everything that's coming into your body will ultimately stay there and cause a health effect. So no huge cause for panic. Just take a deep breath and just try to do your best and follow Scott's advice.
It's good to end on a couple of sort of practical notes for all of us to sort of be mindful about what we can do, but also not overreact until we've got data that tells us we really need to be. So Dr. Scott Coffin and Katie Boston, thank you very much for your time today. It's been a pleasure speaking with you. As I mentioned at the top, this is our 5th episode, and our next two episodes are going to focus on the regulatory aspects of microplastics issues. And so I hope folks will tune in and join us again for those episodes. So but for today, thank you both very much.
We hope you enjoyed today's episode. To learn more about our section, please visit our website at www.americanbar.org/enviro. Please check where you found us for future episodes. Thank you for listening.